Christopher is doing a master's degree in bioinformatics. I'm Zhi Hui, and I come from a biomedical science background, and I'm now studying data science. Politha is in computing and software system, and Matteo is majored in statistics. Together, we combine our different strengths to work on this project. Here's the agenda for today's presentation. We will start with the background and high-level problem, then move on to the pipeline enhancements and integration. After that, we'll briefly share our internship program experience and feedback. And finally, we will wrap up with handover details and next steps. Now, I will hand over to Politha to introduce the background.
Now we will look at the background and high level of the problem. Duplex sequencing is a UMI-based short-read sequencing method that tags DNA/RNA and compare both complementary strands. Helps to eliminate sequencing errors and detect accurately ultra mutation. Because of this high accuracy, this is used in cancer research, studying somatic mutation, studying viral diversity, and etc. Next slide. MultiQC is one of the main tools that is used in this project. It is a reporting tool for bioinformatics quality control that automatically scans pipeline outputs and aggregate results across multiple samples. It generates interactive plots and summary tables, which make it easier to review and compare quality control metrics across samples. In duplex sequencing pipeline, multi-QC is used after calculating QC metrics to summarize them. We will look at this in more detail in the next slide. Let's look at the current duplex QC pipeline overview. It begins with raw sequencing data, where we process FastSQ and BAM file. Then that is feed into duplex metric calculation step, which is a R-script. It is used to calculate efficiency, dropout rate, GC metrics, family stats, and etc. And this R-script produces standardized CSV file, and that CSV file then ingested by MultiQC plugin to do visualizations, where MultiQC plugin produces HTML with plots and bar graph. This is how the basic pipeline look like.
So the existing pipeline had several limitations and did not reach the minimum viable product target. So out of all the metrics, there were only two computed, the efficiency and dropout rate metrics. There was only one installation method through RAMP, which was not very flexible and it was prone to break. There was a first attempt to Dockerize the pipeline, the RepoC calculate duplex metrics pipeline using Docker, but the Docker file was a bit basic and there were missing dependencies, so it wouldn't run. There was no proper testing framework to validate the calculations of the metrics obtained from RepoC. The README documentation of RepoC was quite limited, and the MultiQC plugin generated only a limited set of plots, and there was no validation of the input to check whether the structure of the CSV TSV file was correct. Next slide. So our goal this past semester was to enhance the existing pipeline and reach a minimal viable product. Next slide. This is an overview of what we have done and what we have touched. So we edited the RepoC calculate duplex metrics. We refactored it and we enhanced, we expanded metric coverage and other small enhancements on the CLI. We also edited the MultiQC plugin repo to standardize the output format and include new metrics, new metrics plots. And we also added a unit testing framework, fixed the Docker file, and allowed for other installation methods other than Renv for to grant reproducibility through Docker, or to allow for higher flexibility through Conda. Next slide, please.
Yes, so in the previous version of the duplex matrix calculation REPL, the workflow logic was distributed across multiple scripts. Although this calculate.r script exists, it only contained partial functionality and wasn't fully integrated into the execution path. So most of the workflow control still live inside this cal underscore duplex underscore matrix.r script. In the refactor version, we merge and consolidate the workflow control into a single calculate.r script, and it clearly separated from the pure matrix logic, which contained inside the calculate underscore nanosig underscore function.r. And this improvement helps to reduce redundancy and minimize potential errors. And overall, this results in a clearer structure and a more maintainable foundation for extending matrices. For the CLI interface, we also made some improvement. In the previous version, it only compute 2 matrices by default, and there was no way to select specific matrix. It only support a single input file that makes scaling and customizing the analysis quite limited. So we redesigned the CLI to be much more flexible. We introduced matrix selection through this matrices flag. We enable explicit GC content computation using a reference genome, and we added support for multiple input or directory. And we also allow parallel execution via the cores flag. Overall, this makes the workflow more flexible and easier to use in different research settings.
Now I will walk you through the improvement we made to the MultiQC plugin and its integration. In previous version of the plugin, it took TSV files as input and it only produced metric plots for dropout rate and efficiency. But in the current version of the plugin, we transition into a CSV input file and has automatic header validation, which essentially means we look at the CSV files header for a specific format to confirm it's the right CSV file. Basically, format is sample matrix value which align with the output from R file. And so that made the CSV parsing more robust and seamless, integrated seamlessly. On top of this, we implemented general statistics table, which gives a high-level overview of the key metrics across multiple samples. Additionally, we implemented new bar plots for fraction singletons, total families, GC derivations, and also group plots for GC and family metrics. And finally, we made key bug fixes to the final previous code of the MultiQC plugin, one of which was fixing the PY project TML, which prevented the plugin from installing. And we added more synthetic data for testing. And we also fixed the rounding issue with the plot labels for better visualization.
Yes, in terms of validation, initially we had one standalone script which tested that the output from the calculate duplex metric script matched those in a reference file. However, this was limited in scope of testing, and so what we did, we refactored that into the unit testing structure using the test that package. And so now we can source functions from the calculate and nanosec function scripts and then test them in isolation to make sure that they're working well. In total, we have about 35 unit tests, which test that the functions return valid outputs, and also they work well with edge cases such as invalid outputs. So currently, there are no test failures or the tests pass well. Next slide.
So, as I said before, the previous pipeline was had like the previous group built a Docker file, but it wouldn't build the image because there were missing dependencies. So we added the missing dependencies to the Docker file, and now the image can be built and run. Also, as I said before, the only installation method available was through renv, which is prone to break if the user does not have the same R version as listed in the renv.log file, as some packages might not be available and it would throw an error. So that's why we introduced another installation method through DevTools, which basically takes the whole repo and converts it into a package and lets the user install it as if it was a normal package. And it lets R decide which packages to use, which versions of the packages to use based on the current R version of the user. We also added Conda support because the upstream part of the pipeline also uses Conda, so this allows for continuous workflow across repos. And obviously, we also added Docker, which is the preferred solution when it comes to reproducibility, given that it can be run on any machine and it will reproduce the exact same environment as it was intended from the renv.log file. Next slide, please. So these are our internship experiences and what we have learned and benefited from this experience.
Yes, so for me personally, I've learned a lot. I've gained a lot from this internship. I've learned the basics of software development. I've learned quite about testing in production, and I've gained a better understanding of developing reproducible pipelines. I've also improved my efficiency in R and Python, and also learned quite a lot about teamwork, collaboration, using Git, and also communication in general. I made a small contribution where I converted the validation scripts into the test that structure, and also ran other interns' code and made sure it ran well. Next slide.
So during this internship, my contribution mainly involved reproducing and enhancing the Duplex QC pipeline. I primarily focus on refactoring and improving the Duplex matrix calculation R script and repository structure. This includes extending the matrices beyond the initial 2 matrix version and refactoring the calculation repo into modular CLI tool with more flexible input handling. And I also supported the alignment of output for multi-QC integration. I gained valuable experience from this project. I strengthened my R programming and CLI development skills. I gained deeper understanding of Duplex QC workflow design, and also gained hands-on experience with Git-based collaboration and pull request reviews. Importantly, I also developed stronger teamwork and communication skills.
Yeah, my main focus of this project was to extend multi-QC's metric plots and implement general statistics tables and CSV parsing on top of automating header detection. Beyond code, I gained invaluable skills such as developing strong GitHub workflow, becoming comfortable with branching, pull requests, and collaborative code reviews. Most importantly, I improved my communications and teamwork by coordinating closely with the rest of the team to align on implementation details. And it's been great experience gaining practical hands-on skill with the tool as multi QC. And I also gain experience maintaining a technical diary.
My contribution mostly involved finalizing the Dockerization of Calculate Duplex metrics repo and introducing additional installation methods that would grant more flexibility and reproducibility. Plus, I also updated the README file of RepoC. I gained a deeper understanding of Git and GitHub, and when we had to create branches and make pull requests, and that was very, very interesting and useful. I learned how to use Docker and the importance of using containerized environments to run a reproducible pipeline. I also learned to ask the right set of questions to define the problem and the user needs, such that to make sure that we are both on the same page and before diving into the problem. And I also learned the importance of documenting every single step for traceability through a technical diary in order to get swift support if needed. Next slide, please.
So in terms of feedback and suggestions, I would like to encourage the future interns to ask as many questions as possible. It helps to get a deep understanding of the problem at hand, and also to keep track of their work and document it well, and also make sure they are working with the most up-to-date repo, and also communication between them and their teammates is very key. Next slide. So in terms of handover and where we left off, I would like to have the next cohorts do a full integration of the integrate duplex metrics cachation repo with the multi-C via SnakeMake for better management, and also to automate the end-to-end execution. In terms of documentation and resources, we've provided here links to the different repositories that we worked with, and also links to the technical diaries and the fast track reports that we worked on. We would like to acknowledge both Roland and Marek for the incredible support that they've given us during this internship. You guys have been around to answer our questions timely and give us feedback whenever we needed it. And we also want to acknowledge the previous cohorts, cohort 1 interns and cohort 2, the names listed here. Thank you so much. That is our presentation.
Thank you.
Thank you.
Thank you so much. Really well done. Really, really well done. I think actually I've been doing this for 4.5 years, and that was one of the clearest presentations I've seen of what was done before, what the state was before, and what's next, what we did. What was done before was really, really clear. Really well done. And I also thought as you were doing this, man, this stuff is complicated. This is not straightforward. There's a lot of moving parts here. So from my point of view, really well done. Marek, you are actually the architect behind this. Any comments or questions you wanted to ask?
Yeah, I would just echo those sentiments. I think that was really great how you guys showed the previous state of things and then how you refactored into a newer design. I think that worked really well and it was really clear. Yeah, I just think it was great.
Fantastic. Really fantastic. Well done, everyone. And the time zones must have been a killer as well, I would say, too. So for you all to communicate and collaborate, and it was clear, it was really clear that you were. A really big kudos to all of you. Fantastic.